Okay, I'm going to kick off then. So today we're going to start by an introduction towards the sustainability of lab spaces and where the high impact areas are broadly. And then I'm going to introduce the LEAF program briefly and to show you what it is and to show you some of the actions that we were working on in the LEAF program. Then I'm going to show you some of the areas that we found challenging to address around clinical labs, diagnostic spaces, around sustainability. Excuse me, I'm a bit ill today. And then I'm going to, if okay, hand over to Rob, who's joined us for a brief bit of feedback from his angle because he manages some labs that have participated in the program that I'll share with you today. So to start, what I wanted to put on screen was a bit of background on this topic, which you will see. Oh, got a few more people that are joining. These are the scopes of carbon for anybody who's not familiar with these. I wanted to just show this related to the NHS because it really relates to the high impact areas and the areas that I'm going to talk about for clinical labs. And by the way, for those who are just joining, if you want, put in the chat where you're from, what trust you're from, and you're interested in this topic. It's nice to say hi to folks as we start this network around clinical lab diagnostic lab sustainability. So these are the scopes of carbon. This is how we measure carbon. So whenever we're talking about net zero, and of course we know about the net zero target for the NHS to be for 2040. But these are the scopes of carbon, and carbon emissions is kind of our metric for sustainability. It's the unit that we all reference, even though it's not comprehensive in every sense of the word. There's other impacts we can have on the environment that aren't just measured in tons of carbon and sometimes are hard to convert into tons of carbon. But that being said, for the most part, we are going to stick by these scopes of carbon and use carbon as our reference metric. Now, scopes 1 and 2 are essentially our energy consumption. Scope 1 is our gas, scope 2 is our electricity. And whenever you hear about, this isn't just unique to, our labs or our clinical environments or NHS or wherever, but the majority of our emissions come from scope 3. But majority of our targets, when we talk about these net zero targets, focus on scopes 1 and 2. So scopes 1 and 2 are high impact are the ones because we talk about it, but actually even higher impact is scope 3, which essentially is all of the stuff that we buy, as well as travel. But I think for the importance of today, we're talking about stuff that we buy. A really important message about this because I'm going to bring it up in a second. So energy consumption is important, but all the stuff that we consume is of greater importance. Now, labs that you work in will use in 3 to 5, sometimes as much as 10, but probably around 5 times more energy than your sort of typical office space. And the reason for that high amount of energy consumption in lab environments is because of the ventilation. It's seems like kind of a boring topic sometimes, but ventilation is a high impact area because we have to turn over that air a lot at a higher frequency than if you were in an office space. So a huge amount of energy consumption of these scopes 1 and 2 is going towards your energy consumption of the building of the ventilation systems, and of course also the equipment and things like fridge, freezers, and etc. But scope 3 again, much greater than scopes 1 and 2. Now, I'm just putting this up for interest because I find this kind of interesting and it's relevant, which shows the amount of carbon over time estimated for the electricity consumption in time. Now, what this is essentially showing is that, your electricity that you use in the UK, more renewables are coming online. So, that's why there's a push often to electrify things as opposed to gas. Gas has an associated amount of emissions associated that you can't really escape because it's gas. And when it burns, it releases emissions directly, and that's why it's scope 1. But scope 2 essentially is this electricity consumption, and over time, we'd expect that carbon to go down associated with because you get more wind farms and more use of solar. It's not as fast as we'd like, and to be honest, as we need, to be frank, but this is the planned, if everything goes to plan, which things change. But anyways, this is on a national scale. So your cost of electricity is going up, but the amount of carbon with it is going down. And that's why, for new lab spaces, there's a push for any spaces to electrify things because it's based on the assumption that over time we'll continue to decarbonize our electricity production. So move over to electricity and away from gas is one of the aims of this. Now that's for scopes 1 and 2. Now, these are the emissions of 2 universities, and I'm going to get to your setting in a second, but what I wanted to show you is that these are, research-heavy universities. So there's UCL, and then there's University of Copenhagen. And for both of them, what I found was interesting is that you had relatively small energy consumption figures. So really small energy consumption, gas. Look at it. Look at that, Copenhagen, doing fantastic. Look at how little gas they use. That is, less than we use at UCL, and it's it's essentially just used for heating these days. So a lot of your boilers and your hot water will come from gas boilers today, and so the heating in the building will come from gas, which historically was cheap, but now is less cheap. But that's not the message. The main message here is this huge amount of carbon emissions associated with everything else. So they've kind of broken it up differently than us at UCL, but the point is, I think the takeaway is look at all of the products. And actually, another takeaway of this, I should say, is that waste is not very high on here as well. I'm trying to see if University of Copenhagen didn't really even list waste. Waste for us was 0.1%, which is not to say that recycling is important, but when it comes to the hierarchy of things that we could do that has an impact, recycling is important, but it's not as important as reducing the consumption. But that's really tough in some environments. So this is the NHS, and I just wanted to show this because it gives a similar message, which is that, again, a lot of this stuff is hard to estimate, but the point is that building energy is small. And building energy consumption is around 10%, which looks around not too dissimilar to us in UCL and to other large institutions that are doing research and et cetera. So it kind of checks out. One of the caveats I want to add, one of the why our data here in sustainability is pretty poor overall, is that while we have metered energy consumption, we have metered, water consumption, the rest of this is all kind of guesstimation. Okay, travel is more accurate, but the way that we come up with these figures, it's not guesstimation, it's based on spend. So that's to say that we have different categories of spend, and we say that if you buy furniture, then it produces this much carbon. But not all furniture is the same. The same goes for equipment. With gases, we're getting better because gases are so high impact at estimating these things. But a lot of the stuff that we buy doesn't have good carbon figures associated with it. So we have to go and to look at our spend and then try to retroactively look at the full impact of these areas. And that is not always easy. Excuse me, sorry, coming over a cold. So I just wanted to explain this background because I find it interesting. It's everywhere. It's not just unique to our labs and the NHS, but this is the challenges that we have as a whole for institutions everywhere, is to try to figure out, what the actual emissions are and where we can reduce these emissions. I'm going to get to our setting in a second, but this is, purchasing is this massive area. It's huge, but the truth is that there are no good, real, transparent metrics for the sustainability of what you buy. So it's really tough. And I'm going to talk about how we can address it and maybe open it up to the group. So purchasing is this big area and we have no metrics on it. So if you buy something, they'll tell you about the energy consumption associated with it. You know how efficient your electric vehicle is, or your diesel car. You know how many miles per gallon it gets, but you don't know how much carbon was required to build it. And actually, that can be, as we can see, really impactful. Our consumption of these materials is having a huge impact. We can't even fully quantify it. We can look at how much that car costs, and then we put a value of carbon associated on that cost. But a cheap, large SUV or something will have more carbon associated than a very expensive Ferrari. That's a bad example, but you get what I'm trying to say. Hopefully, is that we have some work to do in this quantification realm. Now, where we'd like to get to, and this is nothing to do with labs, but it's oat milk. If you like oat milk, because it's nice. And what they did is they put in small letters their kilograms of carbon per kilogram. Essentially, they know that their carbon consumption is way less than cow's milk. So they said, let's put it on our box and kind of challenge the milk industry to produce theirs. And that is that is not what all people are doing. So when you buy a lot of materials, you don't have this type of information. Now, you have to be careful because when buying things, it makes it really uneasy on you. And the reason it makes it uneasy, I had a slide, well, I'll just show you some examples, is because suppliers do good and bad, and there's no transparency around whether they're doing bad or good apart from themselves. They are the self-arbiters in this space. Now, this is a company that's doing good, and I wanted to give an example of some good because they're a polyurethane molding supplier for lab supplies, and they've managed to get to net zero for scopes 1 and 2, which is great. And they're going for net scope 3 as well. That's going to be a really tough one, but it's an example of a supplier that's producing materials that's really focused on their sustainability, so that, you know, if they do that, we have to worry less about our consumption of things. They can produce things in more sustainable manners, but they are not unique, but they're a rarity in this world. There's companies like this, and I know you won't use this for your setting. This is more maybe a teaching setting, by the way. I think I heard somebody mention teaching. These are tip washers. They will wash tips in recognition that we use a lot of tips. Sorry. Oh, I should mute that. But they wash tips. We bought this at UCL to pilot it, and you know, if you trust the marketing material, you would save a ton of carbon immediately after several washes. But that's not the truth. The truth is you don't do that. So you have to use it several thousand times to get a carbon saving, which is okay. You know, that you know, sometimes we do go through that volume. But I wanted to put that caveat because what I'm trying to say is that in purchasing, we have to use this critical thinking to say, you know, is something better or worse? Now, this is an example of somebody on the bad, and I always use this example. This is essentially a mouse cage, and I know you don't work with mice, but I just really like the example because it's a single-use mouse cage, and it was marketed as more environmentally sustainable. But most mouse cages, if you work in animal work, they're reusable, they're washable. And so, you know, this single use was being marketed as more sustainable. They said because you run your washers less. So you use your autoclaves and washers less, so you use less energy, focus on scopes 1 and 2 of carbon, but you're producing a mountain of plastic on the other end. And that's what we need to understand better, is what are these impacts and how do we mitigate them? I'm going to get to this in a second because plastics are really important and they're really tough. Now, this is a paper I worked on with a colleague, and it was essentially just showing that reuse is better in terms of carbon. So people often ask, is it worth reusing that mouse cage or that lab supply or whatever it is? Is it worth washing it because it takes so much energy to reuse things? And in short, yes, it is worth it. These are Falcon tubes that we were looking at to reuse them, in which we were able to reuse them. Now, I'm going to give an example, show you a calculator we built in reference to these items, but these are, you know, single-use bio-based tubes. Here, a company was looking at the single-use tubes that we were using and is reducing the carbon associated with manufacturing them. So in a clinical environment, you can't really reuse these things, but you can reduce the amount of carbon required to manufacture them. So how do we do that? Well, that's kind of what I want to talk about. So we've been doing life cycle assessments on some of these materials, which means that we look at the full carbon life cycle of the materials, and then through that, we're trying to understand what, you know, how we can mitigate them. And single-use plastics is a high we use a lot. And we have two ways to affect this. One is top-down, so we can put it in tenders. And one is what a user can actually influence, so what you decide to purchase. And I know that you're limited in some of these senses for this. So what I wanted to start by talking about was the tender language. Now, when our different trusts all do purchasing, we sometimes use different suppliers, sometimes we use the same. But within these tenders, these purchases, we put in language around different aspects. So, you know, quality, price, but we also put some language around sustainability. And I'm going to stop sharing for a second because I want to show you a piece of work that we were doing, which is a calculator to look specifically at this topic. So this is something I was working on with a colleague, and I want to give credit to a researcher at UCL called Isabella Ragazzi. She's an RA who worked with us on looking at trying to quantify the full impact of lab environments. And what we were missing was that full impact of plastics. So this is a calculator that she built. And the idea of this calculator, so if you remember in my slides, I was referencing bio-based Eppendorf tubes. And we were talking about scenarios. So for example, if you were to go for, okay, we could go for different items here. So we could go for, these are different items you might find in a lab environment. And we can say, you can go for these and you can look at basically the carbon consumption associated with using that and then look at an alternative scenario. So for example, you can see here that if you're going to go from a bijou container to a bio-based tube, so in other words, somebody that looks at their manufacturing of their tube, you can reduce your carbon. Now, we're going to refine this because it's not in perfect, it's not in the perfect format yet to be released, but the idea is that we put this out there so that you can use this and you can access this. Now, very quickly, I want to introduce the LEAF program. So the thing that I've been working on is trying to standardize our approach towards sustainable labs. Now, the example I like to give is that we have health and safety standards, but we don't have necessarily sustainability standards. So for example, we have network events like this, so we bring people together and we try to discuss these topics in a format to try to find solutions. And I would argue that we need standards in this space to make it more common practice and to standardize it to do it quickly as well. So the thing that I've been working on for research labs, and I won't talk about research labs much longer, I promise I'm going to go back to clinical, but I just wanted to show you what the tool looks like in reference because we piloted this in your environments. So this is the LEAF tool in its current format, and essentially we can manage different institutions. Sorry. And then we can look at within this, so this is a fake institution, but each lab that would sign up. So for example, if your lab was to sign up in the future, then you would get access to this, which is essentially a set of criteria. And these are actions related to the environmental sustainability of more research or teaching labs. So they're actions that relate to, do you have a recycling bin with tips and advice and links to what other people are doing. And we ask people to provide qualitative answers against this. So say what they're doing so we can understand. So yeah, we can standardize. It's just like health and safety. We want to make sure that people are addressing the right criteria for this environment. And so why don't we standardize it to make it easier for folks like yourselves, because you're all so busy, everybody's so busy. So, so that's the LEAF program. Now, the LEAF program was written, as I mentioned, for research labs and teaching labs. But there's more types of labs, for example, where you all come from. So what we did was we built a new framework, put it on Excel, and I'm going to show you quickly the Excel. And this is what it looks like. Some criteria similar, but again, the idea here was to standardize an approach towards sustainable lab operations for yourselves. So you work in a clinical lab environment, what can you do? And sorry, very sick. But how can we standardize it, and can we get everybody working off the same hymn sheet to make it easier, more accessible, and just make it quicker? And make it as impactful as possible as well. Find the things that we can do within your environment. And that's what brings me to the questions I have for the group. And I'm going to see if Rob also wants to say a quick hello. But just to end on what we did with LEAF, we standardized. We came up with these criteria and we piloted within 7 different trust environments. And the trust environments then, the trusts or labs that were taking part completed these frameworks and gave feedback. And then I did an audit of them either online or in person to try to understand what took place. And that takes us kind of up to today relatively nicely, which is, this pilot has just been completed. And the idea, but I'd love to get your feedback, is that I would take these actions and put them into the tool that I showed you. Now, for reference, I don't know if anybody has come across this, but the Royal College of Emergency Medicine are doing something for A&Es, for ED environments, so emergency departments. And that's something that I worked on. It's a very similar idea, which was come up with a standard framework for them so that they can transmit it out. And that's been launched actually last week. Even if you don't work in A&E, I'd encourage you to check it out because I think it's a really lovely program. I'm biased, but it's kind of what we're trying to do now with your lab spaces. Now, that's kind of where we've gotten up to. The last things I just want to say is that there were 3 areas that I found were challenging for lab environments in terms of coming up with guidance. We found this with the A&E sites and everything, and it's one thing to set a criteria, it's another to give genuinely good guidance on how to achieve it. And there were some criteria that folks had a hard time with. And one of them relates to what I've been talking about, which is purchasing. Now, purchasing is a tricky one. I showed you that life cycle assessment calculator, because within it, what we found, which was really important, was where the plastics are being manufactured, which is not an easy thing to control on a local level. We also looked at how they're being manufactured, but where they're manufactured and how they're being manufactured seems to have a really big impact. Where they're being manufactured relates to the energy mix of the country they're being built in. So if they're being built in a country that uses a lot of coal and gas or something, then the carbon associated with it is very high versus if it's one in. And I don't know how much range we have in terms of these areas, but I wanted to mention it because it's such a high impact area, and I'd love to in the future understand what affects purchasing decisions and how we can influence them in an easy manner that might reduce the carbon emissions associated, because I know you can't reuse everything. I know you can't reuse, there's too much risk, and the risk of a failed experiment is greater than sometimes definitely worth than the worth of a single tube. But equally, we want to be able to affect these things around purchasing. So I had put on here some notes that there was 2 or 3 areas I thought I wanted to mention that when I spoke with the labs, a couple of folks mentioned. One was purchasing. So how do we affect the purchasing, and how is your purchasing affected? And are there any levers or mechanisms we can give guidance on? For example, can we give standard tender language to go in your tenders? Are you able to even influence your tenders? Because that's such a high impact area. The reason I went into that detail around carbon emissions and scopes is because you can see that it's such a high impact area. So how do we affect it? And I have a few ideas, but I don't think they're perfect, and I'd love to hear from yourselves. The other area I wanted to mention was affecting or engaging the number of assays requested from clinicians. And it's one I've heard, it's not my idea, I've heard it from others. And I know that actually A&E, when we were talking with the A&E framework, they were having some issues around finding ways to reduce the number of assays that are mandated when a patient comes in. But it's yourselves that have to do the assays and the tests. So I wanted to mention those 2 areas because they seem to be very high impact. And they seem to be ones that potentially we could find levers for and we could improve the guidance on. I'm going to take a little breath there and stop for a little bit. I want to see Rob, who's on the call today, was involved in managing some of the labs that participated in our pilot. And I just thought would hand over to hear from him because he can speak on the experience of the pilot. Good and bad. I know we got to improve some things, but yes, over to you, Rob.
Yeah, thanks, Martin. Thanks for your help from the IT difficulties earlier. So yeah, I just put together just 4 slides about our experience with LEAF. So I'll just share that if it will let me. Let me know if you can see that.
Yes, can see it now.
Grand, thank you. So we had an interest in environmental sustainability in our department, and I was aware of LEAF and Martin's work at UCL in the academic side, and as soon as I heard that there was going to be this pilot for diagnostic laboratories, we were incredibly keen to sign up. So we formed a small group of keen individuals that we named ourselves the Microbiology Greens, and we set to work on the criteria that Martin put out. And as he said, there were 16 points on the audit schedule, focused around 7 areas, and we focused on low-hanging fruit. Bronze award was I wouldn't say easy to achieve, but they're all things, if you get your heads together, they're all doable. And I agree with Martin about the procurement being the most tricky thing. I'll come on to that. But it's simple things now in laboratories. So making sure that we had a discussion with our trust waste management lead about making sure that we knew exactly where all the waste went, making sure that we were segregating as much waste as possible into clinical and non-clinical, dirty and not dirty. So making sure that we can recycle absolutely as much as possible, and we're not autoclaving or incinerating things that don't need to go down that route, because financially it's much more expensive and it has a much higher carbon footprint. From a people point of view, the first thing was just forming a group and letting know the lab that we have formed a group. So we have a massive notice board that's just outside the main laboratory, where we have a suggestions board. We update people. It's regularly discussed in our team huddles. It's on the regular agenda for our microbiology management group and our pathology management group. We have a slide on sustainability in the pathology induction. So everyone who comes to work in pathology at Langs Teaching Hospitals gets told about the environmental impact of diagnostic labs and what we're trying to do to make it better. Purchasing, the trust has added, and we have added to our SOPs, a point scoring system for the environmental sustainability of the services and the products that we buy. Being completely honest, the NHS is skint, so it ranks quite lowly. So if you have two items that are providing the same service and one has a much better environmental impact than the other, but the worst performing one is much cheaper, we all know which way that's going to go. And that's obviously the beginning of this journey. This has to go in that direction. With regards to equipment, this is where I think we can make quite big gains, because we know that diagnostic laboratories use a lot of equipment that use a lot of energy and a lot of water. So simple things like making sure you switch stuff off when you're not using it anymore. And one of our teams in our lab found that we had a small benchtop incubator that was basically running 24/7, and it only needed to be needed for 24 hours a week, one day. So switching it off for 6 days out of 7 calculated saving the same electricity as boiling a full kettle 51 times during that week. So every week we're saving the equivalent electricity of 51 full kettle boils. So we've adopted this green kettle symbol, which we now have printed, stuck out and stuck on every bit of kit in our laboratory that can be powered off when not in use. It says it's safe to do this. So for example, we contacted bioMérieux because we haven't moored it off, and contacted Brooker rather, and said, can we power down our Mordi overnight? And they said it will affect the vacuum, it will affect the calibration, it's likely to need more work more often. So we made a pragmatic decision that that was a piece of kit that has to stay on 24/7. So I'd encourage you to go around your lab and look at these things systematically. And I say, what's on all the time that doesn't need to be on all the time. IT was very straightforward. Our IT systems are controlled centrally and all of our machines are powered down overnight. But we have gone round and turned down the brightness on every single screen to 25%. And we've had no pushback from the lab teams either. Samples and chemicals were very straightforward because we're a UCAS accredited laboratory, we know where everything is, we've got limited storage space, so we're not ordering more than we need. If we do have a surplus, we have relationships with other labs in our network where we can move stocks around if need be. The ventilation in our estate is very old and very poor. Heating's terrible, ventilation's bad. There's very little we can do about that. But things like safety cabinets and fume cabinets, we have signage on them, which basically means that when we don't need them anymore, they're powered down safely. Again, it's the small things ticking off. Just a couple more things. Martin quite rightly points out that the vast majority, whatever we do in the laboratory is great, and we should be doing it, and we should be taking these good habits home with us as well, and we should be encouraging our teams to bring good habits in with them. But the vast majority of carbon footprint is in the pre-analytical phase. It's extracting the oil, it's making the plastic, it's transporting the plastic around, it's making the tubes and the cannulas and the bottles, and then having them shipped to us. So when we start thinking about doing less, we have to influence our colleagues upstream to talk about inappropriate testing, as Martin touched on in ED. So we have a real interest in diagnostic stewardship, which is still not been precisely defined, but I regard it as basically doing the right test on the right patient at the right time. And you can argue that as we become consolidated into more networks and we get more hub and spoke and more factory-like processing, it's often seen as being easier just to process the sample rather than to stop and think, does the sample need to be processed? And we know from our GIRFT data that we were very poor at rejecting poor quality samples. So we're beginning to look at high volume tests such as swabs and urines. And coming back to the absolute fundamentals, there's a clinical question that's being asked when a test is requested. And there's an analogy that I really wish I could remember where I first heard it, because I can credit them. But it's essentially, doing a test is like picking your nose. You don't do it unless you know what you're going to do with the results. So we all know in our laboratories, we process a lot of rubbish samples that have got very poor clinical information. And I think we need to move our efforts upstream. So effectively, we're doing more higher quality samples, cutting out some of the rubbish that's adding no clinical value to the patient and potentially causing harm and costing us money and contributing to the massive carbon footprint that we have. So I'm happy to hand back to Martin now, if that's okay.
Yeah, thanks for that, Rob. Kind of like the nose picking analogy. Yes. You've heard from us all a bit just now, and there's a few others that are involved in this network from that CSH has put together. I kind of wanted to just see if anybody had any thoughts on some of those topics that we raised, or if you wanted any topics looked at in more detail in the future. I don't know if anybody has any experiences on, or even if there are no experiences and that you haven't seen anything around diagnostic stewardship. Has anybody done anything around this space, or have any good examples, or do they feel like it's an area that we need to investigate more? Or if not, consumption of materials. On this session, I'm going to stop recording in a second. If you like, if you have any topics that you'd like to see covered in the future for one of these networking events, please do put it in the chat. I think there's a few of us involved in this network, and the idea is that we kind of rotate for some networking events to bring folks together to discuss these topics in more depth. So if there's something that you'd like to see discussed, any topics like the ones or one of the ones that we've covered today in more depth, feel free to put it in the chat. But otherwise, many thanks everybody for coming and joining today and for taking the time to input on it. I genuinely really appreciated some of the thoughts and ideas here, taking down a few notes for myself. Feedback is always welcome on the LEAF program, I should say. You know, it's kind of been within UCL, and I'm debating what to do with it, whether that's the right place or to move it out. Yeah, feedback is very welcome. And if you'd like to get more involved in any of these things, or join the network, or get involved in, or do your own event, if you have anything you want to talk about, you'd be very welcome. But otherwise, that's it for myself. And thank you, Rob, also for sharing your experience there. I'll stop recording now, but